Welcome to the deep dive. Today we're getting into some truly breakthrough cancer research. It really changes how we see the fight between tumors and our own immune system. Our mission for this deep dive is pretty straightforward. We want to understand how a tumor doesn't just, hide from our defenses. It actively drains the energy, the stamina from our body's best soldiers, the T cells.
And crucially, how scientists have found a new way to stop that drain.
Exactly. I mean, immunotherapy has been a game changer, right? Especially for something like melanoma, it's offered real hope. But there's this huge challenge.
Hmm?
Why do so many patients not respond? Or why does a great initial response just fizzle out over time? We've all heard the term T-cell exhaustion. The cells know the enemy's there, but they just can't fight anymore. They run out of gas.
And that's the thing, that exhaustion, it isn't passive. I think that's the huge conceptual leap from this research. These findings show us a hidden signal, a molecular signal, that the tumor is actively using to push T cells into that exhausted state. It's not just hiding. It's sabotage, really. It's forcing the cells that are meant to kill it to just surrender.
So the promise here is massive. If you can find and block that specific signal, you could, revive those T cells?
You can revive them, and you can boost the treatments we already have.
So you could turn a therapy that's only partially working into one that's, completely destructive to the tumor. It's about unlocking the immune system's full potential.
That's the goal.
Let's unpack this.
So when we talk about T-cell exhaustion, what we're really describing is a state that develops after chronic stimulation. Think of a long-term infection, or in this case, a tumor that just won't go away.
So the T cells are just overworked?
They're overworked, exactly. They're still there, they recognize the cancer, but their ability to multiply and, most importantly, their ability to kill the cancer cells, it just vanishes. The authors of the study put it perfectly. They're primed, but they're no longer killing.
Which seems like the ultimate biological paradox, doesn't it? Why would your body's best defense system see a threat like a tumor and just decide to put on the brakes? That feels so counterintuitive.
It does, but it's an evolutionary trade-off. That shutdown is actually a kind of emergency brake for the whole body.
To protect us from what?
From the T cells themselves. If they kept fighting forever against a persistent threat, the inflammation they create would spiral out of control. It could cause massive tissue damage, even deadly sepsis. So exhaustion is a safeguard, really. It's just a disaster when cancer learns to use it.
And we already know about the most famous of these breaks, right? The first wave of immunotherapy, it went right after the PD-1 protein.
Precisely. PD-1 on the surface of T cells, that's the main off switch we've known about for years.
And the drugs, the checkpoint inhibitors, they block PD-1.
They do. They essentially release that primary brake. And that's why they've been so incredibly effective, especially for cancers like melanoma. But here's the rub.
There's always a but.
Right. If you only disable that main brake and the tumor is still there, the immune system often just finds another brake to pull, a secondary signal.
So the tumors find a workaround. They evolve resistance. So if PD-1 is brake A, we desperately need to find brake B and C and D.
We do. And that's what's so cool about this new research. The scientists, they weren't even looking for a T-cell brake to begin with.
That's right. They were on a completely different path. They were looking at a molecule called CD47, but on the surface of the cancer cells.
Yeah, for years, CD47 has been known as the don't eat me signal.
The don't eat me signal. So it tells other immune cells, like macrophages, hey, I'm one of you, move along. It's a cloak.
It's a cloak, exactly. They were trying to figure out how the tumor tricks the macrophages, the cleanup crew, and instead they stumbled on how it tricks the T cells, the heavy artillery. They were totally surprised to find that CD47 has this completely separate job on the surface of T cells themselves.
Wow. So that's a huge pivot. We're not talking about tricking a cleanup crew anymore. We're talking about actively disarming your main soldiers.
Exactly. And the quantitative finding was the real eye-opener. T cells, when they're activated and fighting, they have a little bit of CD47 on them.
Okay.
But when they hit that point of exhaustion, because the tumor's been around for so long, they suddenly start expressing CD47 at extremely high levels. It's like a warning light that only comes on when the cell is at its absolute breaking point.
And they proved this in their mouse models. They had mice that were engineered to not have any CD47 at all. And those mice showed much slower tumor growth, which suggested the problem wasn't just the don't eat me signal on the tumor, it was the CD47 on the immune cells.
Right. And then they went one step further. They took it off just the T cells. And when they did that, those T cells got much, much better at fighting melanoma. So this is the big shift, The T cell is an active participant in its own shutdown through this CD47 molecule.
But I guess the critical question is always how well do these models, these very specific melanoma or colorectal tumor models in mice, translate to the huge range of human cancers?
That is always the million-dollar question in immunology. But the mechanism here, it's so fundamental to just how T cells are regulated that it really suggests it could be broadly applicable.
Okay.
And that gets us to the next piece of the puzzle, the missing partner.
Right, because if CD47 on the T cell is the vulnerability, the tumor has to be using something to exploit it. There has to be a partner molecule. Here's where it gets really interesting.
It does. They identified the culprit. It's a big protein called thrombosbondin 1. Let's just call it TSP1.
TSP1.
And this is the protein that the tumor makes. Specifically, metastatic cancer cells produce and secrete a lot of it. So you can think of it as a key made by the tumor that fits perfectly into that CD47 lock on the exhausted T cell.
So the most dangerous cancer cells, the metastatic ones, are the ones dumping this TSP1 key into the environment around the tumor.
Yes, and that key then finds the CD47 lock on the T cell and slam, it hits the exhaustion brake.
That's the mechanism.
That's the mechanism. And they confirmed it so elegantly. They ran the same kind of experiments, but this time in mice that were missing TSP1. And the results were identical to the CD47 experiments.
So the T cells were less exhausted.
Far less exhausted. That was the real eureka moment. Getting rid of the lock CD47 on the T cell or getting rid of the key TSP1 from the tumor had the exact same effect. It proved that this CD47 TSP1 connection is the pathway.
Which means if you can break that connection, if you can just get in between that handshake, you could restore the T cells.
And that leads right to the therapy. We needed a tool to do just that. So they developed something called the TX2 peptide. Now, most people have heard of checkpoint inhibitors being these big antibody drugs. A peptide is much, much smaller.
It's a smaller, more targeted tool for a very specific job.
A very specific job.
Okay.
TX2 was designed to do one thing. Get in between CD47 and TSP1 and block that interaction to stop the tumor from being able to push that button.
And they tested it in mice with melanoma and colorectal tumors. What happened?
The results were really compelling. When they treated the mice with TX2, it preserved the T cell function and it slowed down tumor growth. The T cells were just stronger, they stayed active longer, they produced more cytokines.
And those cytokines are the chemical messengers, right?
Right.
They're how the T cells call for backup and coordinate the attack.
Exactly. More cytokines, like interferon gamma, means you have a T cell that is fully switched on and doing its job. They were way better at actually getting into the tumor and killing it.
But the real excitement, I think, the thing that points to the clinic is the synergy. Using this TX2 peptide alone is one thing, but what about using it with the PD-1 blockers we already have?
That's where it gets really powerful. Think about like a layered defense. PD-1 is the main brake. This CD47 TSP1 pathway is a secondary brake that the tumor uses when the first one is blocked. And the study showed that when they used the TX2 peptide together with PD-1 immunotherapy, they got far, far better control of the tumors than with either drug by itself.
So you block both pathways at once.
You block both. And that's almost certainly the future of immunotherapy, these dual targeting strategies. You block brake A and you block brake B. And you get T cells that can maintain their strength and hopefully avoid developing that resistance that we see so often.
So what does this all mean? This deep dive, it shows us that T-cell exhaustion isn't some passive process. It's not an accident. It's an active signal that can be hijacked. And by finding this CD47 TSP1 pathway, we have a clear, specific target to sort of reawaken the immune system.
And it has just an enormous potential to expand the number of patients that can actually benefit from these therapies. Instead of it only working for a subset of cancers that are sensitive to PD-1 blockers, you now have a tool that could potentially help in a much wider range of tumors. It really tackles one of the biggest hurdles in cancer treatment today.
And that brings us to a final provocative thought for you. We've established that this exhaustion mechanism, it's also a crucial protective system, right, to stop runaway inflammation.
Critical fail-safe.
So while this dual targeting strategy blocking both PD-1 and this new CD47 pathway sounds incredibly powerful for killing tumors, how can researchers be sure that releasing both of those brakes at once won't accidentally trigger a dangerous systemic inflammatory response when they finally test this in people? It seems like the ultimate tightrope walk between unleashing the immune system and causing it to break down completely.